Hello boys and girls, this is Mr. Wong, and this video is about the second part of enzyme. Last lesson we talked about some applications of enzyme. I showed you the video of the fun introduction to enzyme, and we talked about some specific term in enzyme, such as they are made of protein, biological catalase and substrate, and I hope you still remember that. In the video, if you still can recall part of the video, and there should be this funny, very cute enzyme that you can see last time, made of different kind of shape, and they look differently because they are different enzyme. And in the video, I also talk about there are three kinds of properties that you have seen. So if you try to come up 3 words about the conclusion from the second part of the video, there should be 3 words. So the first one should be, of course, just now in the previous lesson we talked about they are made of protein, right? And how about the second one? The second one is talking about the enzyme are specific. Alright, so that means they can't work with the other molecules that they don't belong to. The last one is talking about recycle. That's right. Alright, so in long sentence, that of course you can say in this way, or instead of saying recycle, you can use the word reuse. It doesn't matter, the ideas is the same. Alright, so when you are trying to jot down your notes, I would more prefer you to use some more simple wordings, right? Because you don't actually need to write down the whole sentence, as long as you understand and remember how to represent your ideas. So this is a picture generated from a computer. You can see there are some blue giant like a object here, and also some red object, a cluster of object here, and this is the first scene. And after that, the red object here move inside it get hung by the blue object. So obviously this is showing enzyme, as you can see from the title. Which one do you think is the enzyme? Think about that. If you still remember how it looked like for an enzyme in the video, you will remember the shape of the enzyme. Usually they have a trough like that. Alright, by saying trough, or you may say dent, alright, inside here. Alright, so that things like object like this can go inside the enzyme. So obviously this one is the enzyme, and while this one, do you still remember how do you call that? Substrate. Okay, talking about the specific point from the second point here, I can show you that here from the picture. You can see for different enzymes, let's say this is enzyme 1, this is enzyme 2, they got their own specific reactant. That's because of their active site, alright, is specified for certain kind of reactants. So let's say if you put reactant 2 into enzyme 1, it won't work because it doesn't match at all. Alright, same as if you put reactant 1 into enzyme 2. The geometry shape does not match, so therefore they won't work. So that's why you have to be very specific to use the correct enzyme to match with the correct reactant so that they will work on the active site. For this page, there is only one thing that I want you to look at, is the wording of biology here. Alright, there is a terminology that you need to know how it means and also how to write it down. Alright, so that's the word here. Alright, enzyme substrate complex. So basically, before the reaction happen, this is substrate alone, this is enzyme, and when they are something like combine, when they combine together tightly, alright, we call that enzyme substrate complex. All right, complex means like a combination of this. So in other words, for enzyme specificity, we can say that there is active site on enzyme, and that is the place where the substrate bind, or this is a good word, bind with the enzyme, and when they combine, when they bind together, we call that enzyme substrate complex. Alright, so these three are the very important keywords. But there's one thing that you need to pay attention to in this concept, and that is the active site shape is not same as the substrate shape. Is that correct? Is that correct? Is it really not the same? Let's look back to the previous picture here. In the previous picture, we can see isn't it the same if you look closely? Or we can look at this one. Alright, the shape at this part, isn't it same as this part? Okay, so let's take a look of this example. The shape for substrate and the enzyme itself, we call that complementary shape. The reason why we don't call that, we cannot call them as same shape, that's because of course they're not the same. Alright, so what does it mean by complementary shape then? If you look at this case, this reactant, of course it can try, when they try to approach the enzyme, it can fit into the enzyme active site. Alright, and of course there would be some reaction happen here. Probably in this case, this reactant 2 will break down into 2 or 3. Alright, but if you try to look closely here, maybe you think at the first time that you think, oh, it looks actually the same maybe. But if I try to rotate this around, and when you try to compare them, it is not actually the same, right? So obviously we cannot call them the same shape. Instead, since they can match together without showing any space here, we will call that complementary shape. So I now want you to think about, is there any complementary shape example that you can see in your daily life? Remember, when we say about complementary shape, it means that there could be like two different kinds of object, no matter how they look like, alright, at the back, as long as they could match each other, then we can call that complementary shape, like this one. Alright, I just make it up. Alright, as long as they can fit together here, then we will call that this is a complementary shape example. So I'm sure that you can think of at least there are two kinds of example. So one is this one. Alright, what is this one? Of course, this is the jigsaw puzzle. Alright, and other than that, think about if there's anything other than the object itself. It means, how about the living organism then? By saying living organism, I still remember last year when I talked about complementary shape, some of the students in my class they said, well, in red face, they said their daddy and mommy. And of course, I think what they means is talking about the hand. Alright, you can see the left hand and the right hand. Alright, and that's why your parents can hold their hands together, which they can closely fit together. Alright, so that's the nature, right? So other than the complementary shape example that we just talked about in the PowerPoint, you can go and search tessellation on the Google, and you will see a lot of tessellation pattern that probably you have learned in maths before. Of course, these are some patterns that they can fit with themselves, fully matched together without having any space in between. Of course, this is more kind of superior than the complementary shape itself. Because for complementary shape, just now we talk about, let's say the hand itself, we only require the complementary shape patterns only need to match at one side only, which is the side that they put together. But for like this kind of pattern, they have to match with its own pattern and at all direction as well. But anyway, this is a good example that you can look for if you try to search for some complementary shape pattern on internet. In biology, there is a very classical, well-known metaphor for describing enzyme, which is called the lock and key. So let's have a look of this lock and this key. One of them will represent enzyme, and one of them will represent of course, the other one, which is the substrates. So let's think about which one is the lock, which one is the key. Imagine there are thousands, millions of locks you need to open. How many keys do you need? You only need one key, right? So if you can have one enzyme, well, that's the least amount you need, right? You can help you to have the chemical reaction on the substrate for thousands, millions of times. So right here, the key would be the enzyme, while the lock will be the substrate. Alright, and this is also referring to the recycle properties and also the specific properties in terms of the enzyme itself. Why? Of course, just now we talked about you can use the key many times, so it is recycled just like the enzyme, because you won't throw your keys away once it is used, right? The second thing is about specific. So how is it specific? Do you think you can use your own key to open up Mr. Wong's home store? If that's the case, I would be really scared, because you can always break into my room. Luckily, that's not the truth. And your own key will only be available to open your own door, right? Not even all your stuff, but just your own specific respective door or lock. So that is the same as the specific property in enzyme. Alright, moving on, we will talk about there are three kinds of function in enzyme, and the first one will be saying breaking down of large molecule into small molecules, which we talked about in the previous lesson and also year 7. All those nutrients like starch, protein and fat. And the second one will be the opposite. If you can recall the video that you have seen in certain parts, there is a part saying that there are two glucose, they try to combine together given with the correct active site of the enzyme. And after they combine, they become a new substance called glucagon. So this is one example of how the enzyme to help two same kinds of molecules to form into a bigger molecules here. So when you think of enzyme, how possibility they can have when they work, probably you can only think of either they are trying to make the molecule become smaller or trying to make the molecule to become larger only. So what would be the third and the last possibility then? Not becoming bigger or not becoming smaller. Then obviously it means remain the same size. But if it remain the same size, or how could that be called as chemical reaction then, if this is the same substance, right? So that sounds a bit weird, right? So let's have a look here. The last one is actually called converting. It is actually reordering the structure, so that's why the size will not change. Let's say if you got a molecules which sequenced with smaller molecules called A, B, C link like that, alright, by the bondings here. So now you got an enzyme specific for this, and they try to reorder structure, and this molecules become a new substance. Let's say it looks like B, C, A at last. So this is of course completely new substance, but with the same size or same mass of the previous molecule. Is the same, but the order are different. So of course this is a different substance. So that is the last function that an enzyme could be. It's called convert. So let's talk about more about the detail for each function. So let's say for the first one, which will break down the large molecules into small molecules, I will be very sorry at this point to tell you that what you have learned in year 7 was simplified. So it was kinda a lie to you all. Alright, I'm really sorry that. In fact, what happened when starch is broken down into glucose is a water molecule is needed to break down this starch here. Alright, even with the amylase, and we call this reaction as hydrolysis. Alright, let's put down this hydrolysis. For hydro, it means that something about water. Alright, so that's why we call it hydrolysis here. Alright, but the main point here is well, you don't need to copy all the things here, but what you need to do is try to start to know the reaction itself. Actually, you need to use water to break down the starch. In other words, if we do it oppositely, when we try to use two glucose to form starch, or a few glucose, not necessary to be two. What I call starch is actually a chain of glucose actually. Alright, what will happen is the water from the glucose itself will be removed by the enzyme to build up the starch here to form the bonding. Don't get freaked out by the name here, because even Mr. Wong as a physics teacher would not remember this, so I would not expect you to memorize this. And the example here is actually the reaction happening in the plant. If you recall what you have learned in photosynthesis, what they do is they are going to produce the glucose, right, from photosynthesis. And of course the plants has to survive, let's say survive from the winter, or survive from the like maybe sometimes there's not much light at all, so they must want to store some more energy. But glucose is not a good form to store energy because it would dissolve in water. Instead, what the plants would want to do will be turning them into starch. Unlike human, of course, we as human can turn like the food into fat so that we can use them later on. But for plants, what they can do is only turning glucose into the starch. But anyway, the reaction here should be well known by you, and the new thing is just knowing there is water removed from the molecules. That means the glucose here. Alright, so that the water will be out from the starch itself. And the name for this chemical reaction is called condensation. Alright, let's put down this. Alright, condensation is very familiar to you, isn't it? It's the same as the name when we say the gases form because of cooling down, it turn back into liquid. Condensation, or some people will call that dehydration. Alright, removing the water. Alright, hydrate, right, or hydro is something related to water, and D means remove. So dehydration or condensation can be named as this reaction. Alright, don't copy this. This is not necessary to copy this. As long as you know and you write down the equations here, I'm sure you would know. Alright, and don't get freak out by the name of the enzyme here.